Welcome back to Anatomiqa. In the previous session, we discussed the general introduction, types of muscle fibers, specific features of skeletal muscle, arrangement of connective tissue and muscle fibers, ultrastructure of muscle fibers, and how it is observed under the microscope with photographs and diagrammatic representations. In this session, we will discuss on the histology of cardiac muscle. During embryonic development, the primitive heart tubes mesoderm or mesenchymal cells or myoblasts align in chain-like arrays, forming complex junctions between interdigitating processes rather than fusing into multinucleated cells like in developing skeletal muscle fibers. Adjacent cardiac muscle cells often branch and bind together through intercalated discs, creating tightly knit bundles of cells that produce a characteristic wave of contraction resembling the wringing out of the heart ventricles. Mature cardiac muscle cells are approximately 15 micron meters in diameter and from 85 to 100 micron meters in length, with a cross-striated banding pattern similar to that of skeletal muscle. These cells have one or two centrally located pale staining nuclei and abundant sarcoplasm. Surrounding the muscle cells is a delicate sheath of endomysium with a rich capillary network enabling continuous, involuntary, vigorous rhythmic contractions. The structure of cardiac muscle is same as in the skeletal muscle, both structurally and functionally, except for few differences as follows. Cardiac muscle branch and anastomose with neighboring fibers. Between the adjacent cardiac fibers, there is junctional complex called intercalated discs. Each fiber has one or two nuclei in the center. Mitochondria are more in number. Sarcoplasmic reticulum is not organized. The terminal cisternae is present only on one side of the T-tubule, forming a dyad at Z disc. It is supplied by autonomic nervous system and is involuntary in nature. It does not regenerate after the damage. The most unique and distinguishing characteristic feature of cardiac muscle is the presence of intercalated disc, which is dark staining transverse lines that cross the chains of cardiac cells at irregular intervals where the adjacent cell membranes meet. These intercalated discs represent the interface between adjacent muscle cells in the form of step-like pattern with longitudinal and transverse regions and contain many junctional complexes. When we talk about longitudinal and transverse parts of intercalated disc, it is in context with long axis of the myofibril. The transverse part is oriented perpendicular to the myofibril and longitudinal part oriented parallel to the myofibril. Transverse regions of these step-like discs have many fascia adherents and desmosomes, which together serve to bind cardiac muscle cells firmly together to prevent their pulling apart under constant contractile activity. Fascia adherents are anchoring junctions for actin and connect to closest sarcomere. Desmosomes are present below the fascia adherents, binds to intermediate filaments and prevent separation of cells during contraction. The longitudinally oriented portions of each disc have many gap junctions connecting the cytoplasm of neighboring cells and provides passage of ions between them. These serve as electrical synapses and allow cells of cardiac muscle to act like a multinucleated syncytium as in skeletal muscle, with contraction signals passing in a wave from cell to cell. Diffusion of ions through the pores in gap junctions between individual cardiac muscle fibers coordinates heart functions and allows cardiac muscle to act as functional syncytium. The structure and function of the contractile proteins in cardiac cells are essentially the same as in skeletal muscle. At places, the myofibrils merge with each other and are relatively few in number. As a result, the striations are not as distinct as those of skeletal muscle. The T-tubule system and sarcoplasmic reticulum are not regularly arranged in cardiac fiber. T-tubules are more numerous and larger in cardiac muscle, and the sarcoplasmic reticulum is less abundant. The T-tubule lies opposite the Z line and are much wider. Sarcoplasmic reticulum dilates only on one side of the T-tubule, unlike the skeletal muscle. The T-tubule and one terminal cisterna together is called dyad. Cardiac muscle cells contain numerous mitochondria, which form 40% or more of the cytoplasmic volume, reflecting the need for continuous aerobic metabolism. In skeletal muscle, only 2% is occupied by mitochondria. Fatty acids transported to cardiac muscle cells by lipoproteins are the major fuel of the heart and are stored as triglycerides in the numerous lipid droplets seen in many cardiac muscle cells. Glycogen granules may also be present. Lipofuscin pigment granules are often found near the nuclei of cardiac muscle cells. This is the diagrammatic representation of the section of cardiac muscle. The specific features are they exhibit a cross-striated banding pattern comparable to that of skeletal muscle. Unlike multinucleated skeletal muscle, each cardiac muscle cell possesses only one or two centrally located pale staining nuclei. The cardiac muscle fibers branch. Between adjacent cardiac muscle fibers, there is cell junctional complex, the intercalated disc. Surrounding the muscle cells is a delicate sheath of endomysium with a rich capillary network. This is the photographic picture of the section of cardiac muscle under the low power of the light microscope. The section is stained with routine stain, hematoxylin and eostin to show the features. You can observe the following features in this picture. Each cardiac muscle cell has only one or two centrally located pale staining nuclei, branching cardiac muscle fibers. Between adjacent cardiac muscle fibers, the cell junctional complexes, that is intercalated disc. Surrounding the muscle cells is a delicate sheath of endomysium with a rich capillary network. Another photographic picture of the section of cardiac muscle under the low power of the light microscope. You can pause the video and go through the features. One more photographic picture of the section of cardiac muscle under the low power of light microscope. This is stained with special staining procedure to show the intercalated discs, which is prominently seen. You can pause the video and go through the features. So with this, we have completed the histology of cardiac muscle, which includes the specific features, structure of intercalated disc, and the dyad. The features observed in the photographs of the histological sections of cardiac muscle. In the next session, we will discuss on histology of smooth muscle.